Hello everyone. Good morning. So, a very good morning to all of you. I welcome you all for today's session. So, today we are going to take an important topic in microbiology, that is gene transfers in bacteria, or you can say bacterial genetics. I hope you will find this topic very useful. Okay? So, let's continue with the topic, that is bacterial genetics. Okay, and you get many MCQs also, and in theory exam also. If you're a second prof student, this topic is going to be very important, and if you are preparing for any competitive exam, you are going to get many questions from this topic. This topic is small yet very important. Okay, so let's start it. So humans, this is a human, any cell of the human. This is the nucleus of that cell. How many chromosomes humans have in a diploid cell? So in a diploid cell, total 46 chromosomes are there. Or instead of 46 chromosomes, you can say 33 or 23 pairs of chromosome are there. So pair number 1, pair number 2, pair number 3, 4, 5, likewise, there are 23 pairs. What about bacteria? Now this is a bacterial cell, not a human cell. How many chromosomes are present inside a bacteria? Humans have 46 chromosomes. How many chromosomes are present in a bacteria? Bacteria have single chromosome, not 46. Bacteria have only one chromosome, single chromosome, and that too circular, that too not linear, in the form of the circular fashion. So only one chromosome or single chromosome is present in a bacterial cell, and that too circular. So please understand the structure of the bacterial DNA before understanding the gene transfer in the bacteria. Okay, so the bacteria contain a circular chromosome that contain double-stranded DNA. You can see only one chromosome is there. Okay, now what about the extra chromosomal genetic element? Now this is a bacterial cell. Let me draw. This is a bacterial cell. This is the chromosome of the bacteria, which is made up of double-stranded DNA. It's a circular chromosome. You can see only one chromosome is there. Apart from this chromosome, is DNA present anywhere else? If the DNA present anywhere else apart from the chromosome, it is known as extra chromosomal. Apart from the chromosome, it's known as extra chromosomal genetic material or extra chromosomal DNA. So yes, bacteria have extra chromosomal DNA. Okay, apart from the chromosome, the DNA. Okay, you cannot see the screen. I'm really sorry. Just a second. I hope you can see the screen now. Okay, I forgot to change the scene, I guess. Okay, so I will start from the beginning. Don't worry, I was drawing the diagrams also. Okay, so you can see I was teaching you a topic, an important topic, that is bacterial genetics. Okay, in bacterial genetics, thank you so much, Pardeep, for reminding me that you can't see the screen. Thank you so much. Anyways, let's start an important topic, bacterial genetics. Okay, now to study the bacterial genetics and understand the various ways of gene transfer in a bacteria, you must understand the bacterial DNA. So I was telling you that this is a human cell, this is a bacterial cell. This is the nucleus of the human cell. Okay, inside the nucleus of the human cell, how many chromosomes are present? Can you tell me? So humans have 46 chromosomes, that is 23 pairs. So 46 chromosome or 23 pairs are present here in a human cell. What about the bacteria? How many chromosomes are present in bacteria? In bacteria, only one chromosome is present, and that too circular, not linear. Here you can see the chromosomes are linear, which are made up of double-stranded DNA. Here also it is made up of double-stranded DNA. It's made up of double-stranded DNA only, but there is single chromosome, only one chromosome, and that too circular. Okay, so you have to understand how many chromosomes are present in a bacteria. Only one chromosome, that too circular and made up of double-stranded DNA. It is made up of double-stranded DNA. So this is the structure of bacterial DNA. Got it? Now I want to tell you extra chromosomal genetic element. What do you mean by extra chromosomal genetic element? This is the bacterial cell. Okay, I told you in the bacteria only one chromosome is there. Let's draw that. So this is the one chromosome inside the bacteria, which is present in the nucleus, and that too circular. It is made up of DNA. Is DNA present anywhere else apart from this nucleus? If DNA is present anywhere else apart from this chromosome, it is known as extra chromosomal DNA or extra chromosomal genetic material. So yes, the bacteria have extra chromosomal genetic material. The bacteria have extra chromosomal genetic material in the form of the two things. Okay, number one, sometimes in the cytoplasm, DNA is present. Double-stranded DNA is present in the cytoplasm. It's not a chromosome. It is known as plasmid. This structure is known as plasmid. What does this structure known as? This structure is known as plasmid. It is present in the cytoplasm. It's not chromosome. That's why it is one of the extra chromosomal genetic material. Sometimes this plasmid goes in the nucleus and integrate with the chromosome. It will, once the plasmid go in the nucleus and integrate with the chromosome, it is known as episome. It is known as episome. Now it is integrated with chromosome, but it is not a part of chromosome. It is not. It is extra chromosomal only. It is integrating with chromosome, but it is not a part of chromosome. Okay. So it is known as episome. So plasmids and episomes are same. Both of them are not chromosome. Both of them contain DNA. Both of them are not chromosome. That's why both of them are known as extra chromosomal genetic material. The point is that you have to understand plasmids are present in cytoplasm. And once the plasmid go in the nucleus and integrate, it is known as episome. So episomes are present in nucleus. How many of you got it? So there are two things, plasmids and episome. Are you getting it? Give me a thumbs up if you got it. So plasmids and episome. Can you see this diagram? Both of them are bacterial cell. I hope you can see. Now in the bacterial cell, this is the double-stranded DNA, single chromosome, double-stranded DNA. Okay, you can see here. Now what about the extra chromosomal genetic material? Apart from this chromosome, where is DNA? So if it is present in the cytoplasm, it is known as plasmid. Once it will go and integrate with the nucleus, integrate with the chromosome, it is known as episome. So plasmids and episomes are same. You got it? Everyone got it? Any confusion? Now let's come on the gene transfer in bacteria. There are 4 ways of gene transfer in bacteria: transformation, transduction, lysogenic conversion, and conjugation. Okay, I will be explaining you one by one in detail. Before that, all 4 together, I will draw on one page. So let's start with the first one, transformation. What do you mean by transformation? Listen, this is a bacteria. Okay, this is also a bacteria. This bacteria have the chromosome and this bacteria have the chromosome. You can see I'm drawing different colors to show you the different type of genetic material, the different chromosomes or the genes in the bacteria, right? Now the first bacteria is dead. Yes, the first bacteria is dead. Once the bacteria is dead now, it get busted. It got lysis. So here the DNA is leaked out. The DNA is leaked out. The first bacteria is dead. I'm saying it is bursted. It is just a lysis. So the DNA got leaked out. Now the DNA which got leaked out, it is entering in the second bacteria freely. It is entering in the second bacteria freely without any agency. It is known as freely. The DNA or the genetic material is entering. And you can say it is naked. Naked or free DNA is entering. It do not require any agency. It do not require any transport. And after entering inside, it is having 2 choices. Either make a plasmid or go and integrate with the nucleus and make a episome. So whatever it is doing, ultimately this is a way of gene transfer. So this method of gene transfer is known as transformation. In transformation, one bacteria is always dead. And the genetic material leaked out from the dead bacteria. And it is going in the living bacteria. When it is going in the living bacteria, it is going freely, naked, without any agency. And this is known as transformation. How many of you got it? What is transformation? Do you have any confusion? You can see in this diagram also, you can see this bacteria is shown to you. This bacteria is the living bacteria. Dead bacteria is not shown. The dead bacteria is already ruptured. And the genetic material is leaked out. You can see this is the genetic material. It is leaked out. Once it is leaked out, it is entering in the live bacteria naked. And once it is entering freely naked, after going in the live bacteria, it is having 2 choices. Either it is making a plasmid or it is making an episome. Give me a thumbs up if you got it. Do you have any confusion? So here the word you have to pick is DNA do not require any agency. You can see the DNA is going freely or nakedly, one and the same thing. Freely or naked DNA, no agency, no agency. This is known as transformation. Okay, the first method in which the bacteria is receiving the DNA that is free or naked DNA, the live bacteria is receiving from the dead one. One bacteria is always dead in transformation. So tell me an experiment to prove it. Do we have some experiment? Do we have some experiment to prove it? Yes, there is an experiment. The experiment done by a great scientist. The name of the scientist is Griffith. So we are having the Griffith experiment that proves that transformation takes place. So what does the Griffith has done? What does the Griffith has done? Let me explain you what does the Griffith. So let me explain you Griffith experiment. The Griffith have taken 4 mice. It's a 4 mice experiment. 4 चूहे ले लिए, 4 mice. And Griffith, by doing this experiment, proved that transformation takes place in reality. We will prove that. Okay, listen. To understand the Griffith experiment, you have to understand a bacteria. The name of that bacteria is Streptococcus pneumoniae. Streptococcus pneumoniae. Have you heard about this bacteria? It is also known as pneumococcus. It is also known as pneumococcus, one and the same thing. So either you say Streptococcus pneumoniae or you say pneumococcus. It is of 2 types. It is of 2 types. Let me draw. See the first type. Okay, see the first type, it is having the capsule. And see the second type, it don't have the capsule. Okay, the first type, which is capsulated, you can see the capsule is present. Okay, the capsulated form, you can see the capsule is present. These are known as smooth form because capsule is giving smoothness. These are known as smooth form or S form. Okay, smooth or S. And these are non-capsulated. You can see no capsule is present. These are non-capsulated. No capsule, so they are rough form. Because they don't have the capsule, so they are very rough. Their surface is rough. So call them rough form. Rough form. Rough is R. Rough is R. So we have two types of pneumococcus, S and R. If I use these terminologies directly, S and R, you don't understand. But now you can understand what is S, what is R. Got it? What is S and what is R? And capsule is required for virulence. So smooth and capsulated form are virulent. They can cause pneumonia. They are virulent. But the rough and non-capsulated form, they are non-virulent. They are avirulent. They are avirulent. They do not cause pneumonia. They do not cause any disease. So for causing disease in human, if this is entering in human, it will cause pneumonia and other diseases in human and the person can be dead. If this is entering in any animal or human, it will not cause anything. The person will be healthy. The animal will be healthy because they are avirulent form. So with this basic knowledge, if you got this, you got it? There are two types of bacteria, the pneumococcus bacteria. I am telling you Griffith experiment. To understand the Griffith experiment, you must know pneumococcus bacteria are of two types. What are the two types of pneumococcus bacteria? You can see this one is capsulated. You can see it is having the capsule. And this one is non-capsulated. It don't have the capsule. Okay, capsulated one is the smooth form because capsule is giving the smoothness. It is S form. And non-capsulated one is rough. You can see because they don't have capsule, they are very rough. They are rough, R form. Capsule is required for virulence, for causing the disease. So these are virulent, these are avirulent. If this is entering in human, it causes disease. If this is entering in human, this will not cause any disease. With this basic knowledge, now if you got the two bacteria, let's do Griffith experiment. We are doing Griffith experiment to true transformation. Transformation takes place in reality. We are doing this experiment. Let's take 4 mice. 4 चूहे ले लेते हैं. 4 experimental mice. Okay, let's take 4 mice on the next page. This is mice 1. This is mice 2. This is mice 3. And this is mice 4. Okay, 1, 2, 3, 4. Now you will tell me, in the first mice, I am injected live S. What's going to happen? Is this mice will be healthy or it will be dead? You tell me. Disease hoga ki nahi hoga? This mice will have disease or do not have disease? You use your common sense and tell me. The second one, I'm injecting live R in the second mice. In the third mice, I'm injecting killed S. Killed S. In the fourth, I will tell you later on. First, you tell me the results in the 3. First, you tell me results in the 3 mice. What will happen in the first mice? You use your common sense. You tell me now. What is S? What is S? S is smooth. What do you mean by smooth? This is capsulated one. So capsulated one causes disease. They are pathogenic. They are virulent. So yes, this mice will be dead. This mice will be dead. Okay, because we are injecting a live bacteria inside this mice. A live bacteria, and that too capsulated. The capsulated one are virulent. Come on. I told you they are virulent. They cause pneumonia. They cause disease. This mice will be dead. And we can understand it also. You will say, ma'am, we understood it also. It's not rocket science. We can understand. Okay. And if you take the blood sample of this mice, whatever you have injected, you will get that only. You have injected live S, you will get live S in the blood. You will say, okay, ma'am, we got it. What about the second mice? What about the second mice? In the second mice, you are injecting R. Although it's live, it's a live bacteria. Live bacteria can cause the disease, but it's non-capsulated. The R one is the rough one. Rough one is non-capsulated. It don't have any capsule. It is avirulent. So it do not cause any disease, and this mice will be healthy even after injection. This mice will be healthy. Okay, we can understand it because it is R. R is rough. Rough matlab non-capsulated. It is non-virulent. Capsule is required for virulence. It is non-capsulated. Although it's live, but it do not cause any disease even after injection. So whatever you have injected, you will get that only in the blood. You have injected live R. You take the blood sample. You will get live R. You will say, okay, ma'am, we can also understand this. Till now, everything is good. Everything is easy to understand. Come on, the third mice. Third mice is also easy to understand. The catch will be there in the fourth one. I will tell you. Listen, third mice. Okay, third mice contain S. You will say, ma'am, S is smooth. Smooth means capsulated and it causes disease. It can cause disease. It is virulent, but it is killed. Come on. Can a kill bacteria, you inject a kill bacteria in me? Will I have any disease? You kill any, you inject any bacteria inside me. Although whatever it is virulent, it is very virulent bacteria, but it is killed. So killed bacteria cannot cause a disease, whatever is the virulence. Virulence kitna bhi ho bhai, kill hai na, boss, kill hai na, so it cannot cause the disease. So killed bacteria cannot cause disease in any animal. Although it's S, but since it is killed, it will not cause any disease. So I will write this mice will also be healthy. Hai na? S can cause the disease, but since it is killed, it cannot cause. So this is also healthy. You will say, okay, ma'am, we also understood this. Not very difficult. Okay. And whatever you have injected, you will get the same thing in the blood. Common sense. Killed S. You will get killed S only. Okay. See the second, see the third. Hana, don't see the first. See the second. See the result of the second. If you're injecting live R alone, the mice is healthy. If you're injecting killed S alone, the mice is healthy. And you understood it well. I guess you understood it well. In the fourth mice, you inject together. You mix these two things and inject it together. Okay, the fourth mice. Hana? So I'm injecting the second and third combination. The mixture of second and third. What is the two thing? This and this. The two things together. That is, can you tell me what is it? Say, ma'am, live R and killed S. Killed S. You mix them. You mix them together and inject in the fourth. When live R is alone, the mice is healthy. When killed S is alone, the mice is healthy. So I was expecting here also the mice is healthy because neither live R nor killed S can kill the mice. Live R cannot kill because it is R. R is avirulent. And kill S cannot kill the mice because it is killed one. Killed bacteria cannot kill. So each of them individually cannot kill, but it is an eye-opener experiment. Hannah, we see the fourth mice is dead. Now you tell me the reason why the fourth mice is dead. Can you explain its death? We were expecting it to be live. We were expecting it to be healthy because individually each of them is healthy. When we are combining them and making a mixture and injecting, we are still expecting the mice should be healthy. But this mice is dead. This mice is dead. Can you explain why this mice is dead? Who has killed this mice? Neither live R can kill because it is R. R cannot kill. Nor killed S can kill because killed bacteria cannot cause the disease. Now, who is killing this mice? If you take the blood sample of this mice, again an eye opener, you will get live S. Come on, we have not injected live S. We have not injected. We have injected live R and we have injected killed S. Ye kahan se aaya? Live S. From where? The mice blood has live S. From where? From where the mice is getting it? Huh? You are getting my point, what I am saying? So what happened actually inside this mice? This killed S is dead now. It leaked out. It is bursted. The killed bacteria just bursted. They show lysis. So the killed S bacteria got bursted. And the genes from this bacteria leaked out and enter in the live R. After going live R, they go and integrate with the live R. The genes of the killed S. So the killed S have one of the gene for the capsule. Capsule का भी कोई ना कोई gene होगा. So one of the gene for capsule. So that capsule wala gene, the gene responsible for capsule, it is leaking out and going in the live R and integrating. So live R start making the capsule and it convert into live S. And that is causing the death of the bacteria. In this way, we are proving transformation. First, you tell me how many of you got it. The persons, all those persons who are live and you got it, give me a thumbs up. Or else, if you have any doubt, write down specifically. You want me to repeat it? What is happening in the fourth mice? Or you got it? Kindly respond. So Shahrukh, all the notes are available on a link, on all my YouTube sessions. In this session also, you will get a link which is pinned in the chat. Okay, if you click on that link, many of the notes are available. Okay, and all these notes I'm going to provide you together also. So can you respond? You get it or you didn't get it? Give me a thumbs up if you got it. Can we go ahead? So this is Griffith Experiment. The same experiment is shown to you here. You have to understand it like this. So we are using 4 mice. In the first mice, we are injecting live S. So the mice is dead. We can understand. The second, live R. The mice is healthy. The third is killed S. Still the mice is healthy. You are combining the two. Second and third. You are using a mixture. But this mice should be healthy because individually this is healthy. This is healthy. Okay. But here the mice is the dead. If you are using the combination, the mice is the dead. Okay. Can we go ahead? So this is Griffith Experiment. Okay. You can understand why it is dead and you have to explain it. So this is transformation. Okay. The first method in which one of the bacteria is dead. Okay. Now let's come on transduction. The second one. I will draw two together. Transformation versus transduction. So you will have a better understanding. So first is transformation. Here we have 2 bacteria. And now I am trying to draw the transduction. Here also we have 2 bacteria. Okay. I will use different colors to show you the genetic material of 2 different bacteria. And I am using red and blue. In the first case, the first bacteria was dead. I told you here the first bacteria was dead. Okay, so it got bursted. The first bacteria got bursted. Once it got bursted, the DNA leak out and it goes freely in the second bacteria. Okay, so here the word to be picked up is free or naked. And we have already studied it. This method is known as transformation. My point is that here DNA do not require any agency. It is going naked by itself. Okay, in the second, we require an agency. Hana, in transduction, we require agency. The agency is a bacteriophage. Who is a bacteriophage? Bacteriophage is a virus. What is a bacteriophage? If you don't know what is bacteriophage, it's a virus which infect bacteria. Virus is infect human. Hana, humko virus infect karte hain, we have diseases. Hana, viruses also infect bacteria. So such a virus which infect the bacteria is known as bacteriophage. It's a virus. Hana, so this is the DNA of the bacteriophage. So this bacteriophage is entering in the first bacteria. After entering in the first bacteria, it is taking a small portion of DNA of the first bacteria with it and listens of the first bacteria and coming out. And after that, it is entering the second bacteria. It is entering the second bacteria. So whatever taken from there, the first bacteria, it is transmitting to the second bacteria. So basically, I mean to say this is acting as an agency. The bacteriophage is acting as an agency, which is doing pick and drop service. It is picking the DNA from here and dropping it here. So I call them Swiggy. I call them. Do you have Swiggy or Zomato services in your city? So Swiggy, Zomato, what does these people do? What does these people do? They just pick and drop. So pick from one place and drop it to other place. So it is the agency which is doing the transfer. It is not free DNA which is going from here to there. It is the agency. It is the agency which is doing the transfer. So whenever the agency is bacteriophage, it is known as transduction. So please clarify your doubts, please. So transformation, here the free DNA is going from one bacteria to another, free or naked. The first bacteria is dead. And here in transduction, you require an agency that takes the DNA from one bacteria to another, that is doing the picking and dropping service. Okay, so the name of the agency here is bacteriophage. Say yes if you all got it. Say yes if you all got it. Do you have any confusion? So this is transduction. Griffith experiment is for proving transformation, not for transduction. For transduction, we don't have any experiment to prove that. Okay, we don't have an experiment, any specific scientist to prove that. So that is, we are done with transduction. What is transduction? We require an agency. What is the name of that agency? Bacteriophage, which is a virus, which is taking a portion of DNA from one bacteria and giving it to another bacteria. So this is known as transduction. This is known as transduction. So here, from one bacteria, this is one bacteria. Okay, this is another bacteria. So this is the DNA of one bacteria. This is the DNA of another bacteria. Now the first bacteria also have plasmids. Okay, and this is the bacteriophage. So my point is that the bacteriophage can transmit the chromosomal from here to there and extra chromosomal also from here to there. And it is an agency. It can transmit the chromosomal DNA also and extra chromosomal DNA, that is plasmid also. It is the best way which is used in genetic engineering nowadays. And you know, nowadays we are using genetic engineering in multiple human benefits. So it is the best way we can do the transfer. It is a vehicle. It is simply a vehicle. It is used for chromosomal as well as extra chromosomal genetic transfer. You can see the same. So this is the first bacteria you all can notice. This is the bacteriophage which is entering in the first bacteria, infecting the first bacteria, causing the infection, taking a small portion of DNA and coming out. After coming out, it is entering the second bacteria. What is taken from here, it is transmitted to here. So this is bacteriophage. I hope you all got it. I hope you all got it. So let's come on the third. So till now we have seen two methods of bacteria gene transfer. Transformation, for which we have done a Griffith experiment also, and transduction. Now coming on the third method. The third method is lysogenic conversion, also known as temperate cycle. Let me explain. Okay. So let's compare the second and third. What is the second method? The second method is transduction. And the third I'm teaching you right now is lysogenic conversion. You have to be understand the differences between them. Okay, you have to be very specific. Okay, so here also two bacteria are there. Here only one bacteria is there. Okay, listen. So here is the DNA of this bacteria and here is the DNA of this bacteria. Now in transduction, I already taught you. In transduction, we require an agency. That is bacteriophage. That is bacteriophage. That is doing pick and drop. Taking a small portion from here and transmitting here. You can see the red color will reach here. The red DNA of this is reaching in the second bacteria via bacteriophage. So my point is that bacteriophage is doing pick and drop service. Pick and drop. Please learn this word. In transduction, you already understood. In lysogenic conversion also we have a bacteriophage. We also have a bacteriophage here also. You will say, ma'am, then what is the difference between transduction and lysogenic conversion actually? Here also you are saying we are having bacteriophage. Yes, we are also having bacteriophage here, but we don't have two bacteria. We have only one bacteria. Here we have two bacteria, na? Here the gene transfer taking place between two bacteria, bacteria number 1 and bacteria number 2. Here we have only one bacteria. So basically bacteriophage is coming and giving its own DNA. Can you see I'm using purple color? It will give its own DNA to this bacteria. So it's the phage DNA which is entering in the bacteria and integrating with the bacteria. It is not two different bacteria. So here bacteriophage is not doing pick and drop service. It is not Swiggy, it is not Zomato. The bacteriophage is giving its own DNA to the bacteria. And this is known as lysogenic conversion. How many of you got the difference between transduction and lysogenic conversion? First you tell me. First you tell me. Okay. So gram-negative bacteria, definitely I'm going to launch that because many requests I'm getting from multiple students throughout the globe. I'm getting the request. Please take a session on this topic, this topic, this topic. So I'm going to get all the sessions gradually one by one on the YouTube. I'm going to schedule it based on the demand. Many students are demanding many topics. Okay. So I'm just... If you have any topic in your mind and you find it difficult and you want me to take it on YouTube and schedule it for YouTube and take it live, I will definitely going to schedule it. Just text me that topic. Okay. So in the end of the session, I will give you a contact number on which you can send a WhatsApp message. So we are making a list and based on the priority, we are just scheduling them one by one. So don't worry, whatever topic you want, we will gradually take it on the YouTube. But maybe it will take some time, but gradually we will take it. Don't worry. Definitely we are going to take it. Okay. So let's continue. Okay, so you got it. So I taught you 3 methods. How many of you got the difference between transduction and lysogenic conversion? Transduction and lysogenic conversion. How many of you got the difference? In transduction, we have 2 bacteria. Okay, so the phage, the bacteriophage is acting as a agency. It is doing pick and drop service. It is picking the DNA from one and transmitting whatever taken to another. Uska apna kuch lena dena nahi hai. The bacteriophage is not contributing its own DNA. It is just taking from here a parcel and delivering the parcel to the other bacteria. That's it. So this method is known as transduction. But lysogenic conversion, the phage own DNA is given to the bacteria and that is integrating with the bacteria. Here we have only one bacteria. So here in transduction, the transfer takes place between 2 bacteria. But in lysogenic conversion, the genetic transfer takes place between the phage and the bacteria, not between 2 bacteria. You have to understand this small concept. I hope you all got it. Okay, I hope you got it. Can we go ahead? Can we go ahead? This is lysogenic conversion. So lysogenic conversion, the definition is phage DNA is integrating with the bacterial chromosome. So and the combination which is formed, it is known as prophase. The best example is diphtheria. Have you heard about the bacteria? The name of the bacteria is Corynebacterium diphtheri. Have you heard about it? There is a bacteria. The name of the bacteria is Corynebacterium diphtheri. It is a bacteria. It causes a disease which is known as diphtheria. Now this bacteria, Corynebacterium diphtheri, this bacteria is of 2 types. Listen. This bacteria is of 2 types, like pneumococcus only. It is of 2 types. You can see. So here, this is the nucleus, the chromosome. Here, this is the nucleus, the chromosome. Now the bacteria which is having tox gene can produce toxin. Tox gene is responsible for production of toxin. And toxin is responsible for causing diphtheria. So because of the toxin only, it can produce toxin and it can cause a disease, diphtheria. So this one is pathogenic or toxigenic. Either call this toxigenic or called pathogenic or called virulent. One and the same thing. Because it is producing toxin, it is pathogenic, it is virulent. It is virulent. I mean, it causes diphtheria. If it enters in human, it will cause diphtheria. The other one, it don't have toxin. It don't produce toxin. Because it don't have toxin, it don't produce toxin. Since it don't produce toxin, it is non-toxigenic. Okay? Non-pathogenic. Okay? And non-virulent or avirulent. Whatever you say. Non-virulent or avirulent. So if this one is entering in human, it is causing diphtheria. But if this one is entering in human, it do not cause diphtheria. Got my point? So Corynebacterium diphtheri is of 2 types. I hope you all got it. Now, is there a way? I'm asking you a question. Is there a way to convert non-toxigenic to toxigenic? Is there any way? Can I convert the non-toxigenic strain to toxigenic strain? Say yes or no. You will say, ma'am, it's a simple funda. If this will get tox gene from somewhere, koi isko tox gene de de. If this bacteria is getting the tox gene from somewhere, it can get converted. But who will give the tox gene? So there is a phage. The name of the bacteriophage is Beta-phage. It's a virus. The name of this bacteriophage is Beta-phage. Learn the name. It's a MCQ. Beta-phage. It is having tox gene in it. It is having tox gene in it. Okay, so this beta-phage is coming and infecting this bacteria, providing the tox gene. Once it will receive the tox gene, it will form the toxin and it will convert from non-toxigenic to toxigenic. So if you want to convert the non-toxigenic strain to toxigenic strain in Corynebacterium diphtheria, you require lysogenic conversion. In lysogenic conversion, you can see this is the Corynebacterium diphtheria, don't have tox gene. So it is non-toxigenic. So here a bacteriophage, the beta-phage is coming and giving the tox gene. Once it receives the tox gene, not as toxigenic, it will produce the toxin, it is pathogenic. Say yes. Say yes, everyone. So this is lysogenic conversion. So in transduction, the phage is acting as a Swiggy, as a Zomato. It is doing pick and drop service, taking the parcel from one place and giving it to other place. But here phage is giving its own DNA to integrate. How many of you got it? How many of you got it? This is the third method. So till now, I taught you three methods. What are the three methods till now I taught you? The first is transformation. The second is transduction. And third is lysogenic conversion. Most of the students have confusion between transduction and lysogenic conversion. Let's revise. And the fourth one is still there, conjugation. So let's revise the first three, and on the same page, I will draw the fourth. So on this page, I'm drawing all four methods of gene transfer of bacteria. Please have a look. Okay, please, everyone on the screen. I want everyone on the screen. So the first method is transformation. I'm going to draw here. The second is transduction. I'm going to draw here. The third, lysogenic conversion, I require only one bacteria. Okay. And in the fourth one, conjugation, I will draw it here. Okay. Let's draw the four method one by one. Let me show you. First method is transformation. Okay. The second method is transduction. Okay. The third one is lysogenic conversion. And the fourth one is conjugation. These are the four methods by which the genes of one bacteria can be transmitted to other. Okay, so gene transfer in bacteria, the bacterial genetics which I am teaching you right now. Okay, you got it? So let me draw the DNA. Okay, let's draw it like this only. Let me draw the DNA in all these bacteria with different colors so that you can have an understanding. Okay, now let's start one by one. Let's talk about the transformation. In transformation, what is happening now? The first bacteria is dead. Yes, the first bacteria is dead. This bacteria is dead. It got busted. It got lysis. Once the dead bacteria, the first bacteria is dead. Okay, the one bacteria is dead or killed. It is busted. It is lysis. So DNA got leaked out. So the free DNA is traveling and entering in the second bacteria. So no agency is required. Here the DNA is free. Here the DNA is naked. So free or naked DNA entering from one to another bacteria, this method is known as transformation. This method is known as transformation. Say yes if you got it. Say yes if you got it. Now coming on the second method. In the second method, you require an agency. Here we don't require any agency. The bacteria, the DNA is going walking from one to another. The first bacteria is busted, it is leaked and walking, simply it is going in another bacteria and it is integrating in the another bacteria, making a plasmid or episome. But in the second method in transduction, we require agency. The name of the agency here is bacteriophage, which is a virus. Bacteriophage. It is a virus. So here the bacteriophage is acting like a Swiggy, along like this, like Zomato. Mainly it is doing pick and drop. So pick and drop service. What do you mean by that? So this bacteriophage entering the first bacteria, picking a small portion of DNA from this bacteria, which is known as packaging error, and it is coming out. And after that, it is infecting the second bacteria. Whatever taken from the first bacteria is transmitted to the second bacteria. So the red DNA taken from the one, it is transmitted to the another. It is pick and drop. Simply pick and drop. So this method is known as transduction. In transduction, there is an agency. The name of the agency is bacteriophage. Here we don't require in transformation no agency. Here the bacteria, the gene transfer take place freely or by naked DNA. Free or naked DNA. Here the agency is bacteriophage. Now coming on lysogenic conversion, the third method. In lysogenic conversion also, we require an agency. Here we have only one bacteria and here also we have bacteriophage. So what is the difference between here and here? Here also we have bacteriophage. So here phage own DNA is given to this bacteria that will go inside and integrate with the bacteria. Phage own DNA. Phage own DNA. Okay. It is not pick and drop. It is not pick and drop. Here, in transduction, it was pick and drop. Here, there is no pick and drop. The phage own DNA is given to the bacteria. And finally, conjugation. Finally, the fourth one, the last one, conjugation. Conjugation is known as sex in bacteria. It is known as Sachdev, the other name. It is known as mating in bacteria. So basically, in the first, second, or third, the bacteria are not coming physically closer. Here the free DNA is leaking from one to another. Here the phage is the agency, taking from one to give to the other. Here also phage is giving its own DNA, but here the bacteria will come physically in contact. That's why it is known as sexduction, sex in the bacteria, or it is known as physical contact or mating in the bacteria. So the two bacteria come closer. They physically attach with each other. Now they will form a tube between them. They will form a tube between them, which is known as conjugation tube. Okay, it is known as the conjugation tube is made up of pili. The bacteria have pili, pili or fimbriae. With the help of pili or fimbriae, they will form a tube between them, and via this tube, the gene transfer takes place from one bacteria to another. Now imagine this bacteria have a plasmid. It is transmitted to this bacteria via this. So this is known as conjugation. So conjugation, it is the physical contact between the two bacteria. So that is the fourth and the last, that is conjugation. How many of you got it? How many of you got it? So I taught you the 4 methods. Conjugation, I will give you the details. Okay. Transformation, here no agency is required. The bacteria is going, the DNA transfer takes place by free or naked DNA. In transduction, there is an agency. The name of the agency is bacteriophage. In lysogenic conversion also there is a bacteriophage, but it's phage own DNA given. And in conjugation, it is the physical contact between the two bacteria via a tube. The name of the tube is conjugation tube. How many of you got it? Thanks a lot for your feedbacks, for your comments. I'm really happy to see all of them. Thank you so much. Thank you so much for your genuine feedback. And I will try my best to deliver the best content in the medical world to all the medical students. So whatever topic you find it difficult, kindly text me. And you want me to take it on YouTube, I will definitely going to schedule it. That takes my words. Okay, maybe time will take, but one by one, I'm scheduling all the topics on the YouTube. Okay, live only. Okay, thanks a lot for the feedback. Thanks a lot. The only thing I want from your side is your success in your profession. Okay, whatever exam you are targeting, whether it is a prof exam, whether it is NEET PG, FMGE, INICT, USMLE, PLAB, or whether you are a paramedical student, some nursing or dental student, whatever exam you are targeting, you should get succeed in that. That is the only gift, written gift you can give it to me. Anyways, let's proceed. So I'm giving you the details of the conjugation. I was teaching you the 4th method. So I already told you what is conjugation. What is conjugation? It is physical contact between the two bacteria. It is mating between the two bacteria. It is known as sexduction. So see the two bacteria. Can you see the first bacteria and the second bacteria? They are coming closer. They are making a tube between them. They are making a tube. Via this tube, the genetic transfer between the one and the second bacteria takes place via this tube. This is known as conjugation. Now there are 2 types of conjugation. You have to learn the 2 important conjugation. The first is F factor, F-plasmid, and second is R-factor. F for fertility and R for resistance. Learn the full form. F for fertility. It is F-plasmid, F-factor, also known as fertility factor or sex factor. Okay. And second is R. R is resistance. Let me explain you. These are the 2 important types of conjugation. Let me discuss one by one. The first is the F factor. Let me discuss F factor. What is F factor? Listen, in bacteria also there is male and female. Okay. Can you see the first bacteria? Bacteria A. And can you see the second bacteria, bacteria B? What's the difference between them? See their structure. See the first diagram, only the first diagram of both of them. Can you tell me the difference? In this bacteria, this is the nucleus. In this bacteria, this is the nucleus. So nucleus or chromosome is present in both of them. But the first bacteria have an additional thing, F-plasmid. It is one of the extra chromosomal DNA. F-plasmid, F for fertility. It is having. So I will call it F-positive bacteria. F-positive are males. I will call it a male bacteria. The second one don't have F-plasmid. So I will call it F-negative bacteria. Or I will call it a female. I will call it a female. So they will come physically closer. They will make a tube between them, which is known as sex tube, or conjugation tube. Now the male bacteria, F-positive bacteria, will replicate the plasmid. One copy is kept by himself and another copy is given to the partner. The second copy is given to the partner. So at the end, both have F-plasmid. So at the end, both are F positive. So my point is that, see in other words, see. My point is that when we start the conjugation, one is F positive, one is F negative. That is, one is male, one is female. At the end of the process, both are F positive, both become males. So after the sexduction, both become male. So when they start, one is male, one is female. But when they end, after the sexduction, after conjugation, both become male. So conjugation is providing maleness. So here, this is the donor, this is the receiver. The donor is the male, it's F positive. The receiver is the female, it's F negative. So it is fertility factor. How many of you got it? How many of you got it? So I told you F positive is the male, that is donor, and F negative is the female, that is recipient. Okay. And after conjugation, when we start, one is F positive, one is F negative. And when we end, both of them become F positive. Okay. You have to understand it. Second is R factor. R is resistance. R for resistance. See. See, this is a bacteria. This is a bacteria. Imagine this is a bacteria and it is present in my patient's body. This is a bacteria, it is present in my patient's body. I'm a doctor. I want to treat my patient. I want to cure my patient. But to cure my patient, I have to kill this bacteria, which is present in my patient's body and causing a disease. So I want to treat my patient. I want to cure my patient. So I have to kill this bacteria. So I have to murder this bacteria. Then only my patient will get treated. So to kill my bacteria, basically I am murdering this bacteria. So basically all doctors are doing the murder of the microorganisms. Okay, so we want to treat our patient. That's why. Okay, so we are murdering this bacteria. Okay, we are killing it. Then only our patient will get treated. But nobody likes murder. If someone is murdering me, I will do the defense. I will shout, help, help, and I will do the best I can do to save myself. Okay, in the same way, if you are killing a bacteria which is present in your patient's body, this bacteria is present in your patient's body. This bacteria do not want to get killed. Nobody wants to get killed. Bacteria want to live. Okay, so bacteria will produce a R plasmid in that. R for resistance. Okay, R plasmid. Some bacteria will produce R plasmid for the particular antibiotic. So to kill this bacteria, you are giving an antibiotic. For example, you are giving tetracycline, you are giving cephalosporin, you are giving penicillin, you are giving any antibiotic. So for that particular antibiotic, it will produce the genes that will resist the action of that antibiotic. So this is R. So R plasmid is good for the bacteria but bad for human. The bacteria which is having R plasmid, that is not killed by that particular antibiotic. So bacteria will survive. So it is good for the bacteria but not good for us. Our patient will not get treated. So this R plasmid is good for bacteria but not for humans. So you're getting my point what I'm saying you. Now listen, all bacteria do not have this R plasmid. Listen, there are two friend bacteria. Best friends forever. Okay, this is bacteria A and this is bacteria B. See the first diagram. Okay, what's the difference between the two friends bacteria? See this bacteria. It is having a nucleus. Okay, see the second bacteria. It is also having a nucleus. Okay, what is the difference? The first bacteria have an additional R plasmid. Can you see? This is R plasmid. Second do not have that. Okay, I will call it a R positive bacteria because it is having the R plasmid. And I will call it a R negative bacteria because it doesn't have it. So on the R plasmid, I can see various genes. Don't learn the genes. It can differ from bacteria to bacteria. Here T for tetracycline, S for sulfonylurea, maybe S for cephalosporin, C for cephalosporin, sulfonamide, multiple antibiotics. So these four antibiotics, if you give tetracycline, S sulfonylurea, cephalosporin, and sulfonamide, if you give any of them, this bacteria is not killed because it is having resistance. It has developed the resistance for these four antibiotics, or it can be more than four, multiple. It has developed multiple. So for that particular gene is present on that plasmid. If you give any of them, this bacteria is not killed. This bacteria is not killed by any of the four because it is having genes for these four. Don't learn the name of the genes. It can differ. It can be for penicillin or any antibiotic. Okay. Now this bacteria don't have it. Now if you give these four antibiotics, this will get killed. This will get killed. So B is saying to A that I'm your best friend. Now once the tetracycline will come, cephalosporin will come, any antibiotic will come, you will survive, but I will kill. So it is not a good thing. We are best friends. I should also survive. So can you give that plasmid to me also? Can you give that plasmid to me also? So the A bacteria is saying to B, yes, we can do so. So come closer, we will make a tube. So the two bacteria are coming closer. They are making a tube between them. The Sachdev they are doing. So they are mating with each other. So the first bacteria, the A bacteria, is replicating the R plasmid. One copy is kept by himself and one is given to the best friend, the partner, the first. Now at the end, both become R positive. So once we started, one is R positive, one is R negative, but the end both become R positive. Give me a thumbs up if you got it. The same which was occurring in the F plasmid. At the start, one is F positive, one is F negative. At the end, both become F positive. Here also, when we start, one is R positive, one is R negative. But at the end, both of them are become R positive. Okay, say yes if you got it. Okay, so this is a transfer of resistance. This is a transfer of resistance from one bacteria to another. Okay, you got it. So this is R factor. So we are done with the 4 ways of gene transfer in a bacteria. How many of you got it? What are the 4 methods? Transformation, transduction, lysogenic conversion, and conjugation. Transformation, here the DNA, free DNA or naked DNA is moving from one bacteria to another. The first bacteria is dead. Here, the agency is bacteriophage. The bacteriophage is doing pick and drop service. It is acting like Swiggy and it is taking the DNA of one and transmitting it to another. In lysogenic conversion, here also we have bacteriophage, but phage own DNA is integrating with the bacteria. And in conjugation, it is the physical contact. A tube is formed. I hope I'm not drawing it again and you got all the 4 methods. Say yes if you got it. Say everyone yes. Now in the end, before ending the session, I would like to explain you PCR. It's ultra important topic. PCR, polymerase chain reaction. It also come in bacterial genetics. What is polymerase chain reaction? What is chain reaction? Chain reaction is something that requires initiation. Once you have started, the reaction will continue by itself for indefinite. Such a reaction is known as chain reaction. You have studied the chain reaction in chemistry in childhood, whenever you were a kid. So everyone knows what is a chain reaction. I'm teaching you here polymerase chain reaction, that is PCR. What do you mean by PCR? It is a DNA amplification system. You can call it a Xerox machine. Xerox machine. Have you heard about Xerox machine or photocopy machine? So we put one copy in the Xerox machine and we just put a number that we want 1,000, we want 10,000, we want 100 copies. So the Xerox machine is Xeroxing that many of copies. So the same thing is happening in the PCR also. It's like a Xerox machine. You are getting my point? So basically, just suppose this is my patient and my patient is having certain infection. Okay, so I'm taking the specimen from my patient. The specimen can be anything. It can be blood, it can be urine, it can be anything. It can be serum, it can be saliva, it can be CSF, it can be bone, it can be anything. Now I'm suspecting my specimen contain the microorganism. My specimen, I'm suspecting, I'm not sure. But the count is very less. Microorganism is very less in amount. It's very less in amount. So I cannot diagnose it because it is very less. Although it's present, but it's very minute, maybe 10 to the power something, minus 5, minus 6, that many of grams. It's very less. So before diagnosis, I want to amplify it. I want to amplify. I want to make multiple copies. I want to Xerox it. So that's why I require a PCR here. That's why. So here in PCR, the particular gene of interest of your microorganism can be amplified to any extent, to any number of copies. It can be a bacteria, it can be a virus, it can be a fungus, it can be a parasite. Any of the gene can be amplified. You're getting my point? So it is a DNA amplification system where large amount of DNA can be produced from small amount of DNA. So it amplifies DNA. It was discovered by Carey Mullis and he got a Nobel Prize for that. It produced a revolution in the microbiology. So that is PCR. No. Okay. Now there are 3 steps in PCR. You have to understand the 3 steps of the PCR. Okay. Now this is the double-stranded DNA. Can you see this is double-stranded DNA? I want you to appreciate this double-stranded DNA. Okay. This is PCR machine. This is known as thermal cycler. It is PCR machine. So take your specimen and put it inside this machine. It's nothing is manual. Everything is automated. So just put your specimen inside this machine and switch on. And you put how many copies you want. Put it in the software. How many copies? You want 1,000 copies, 10,000 copies. How many copies you want? Put that number. After that, it will automatically stop. Okay, till that it will continue. Okay, now put your specimen here which contain a small amount of DNA. A small amount of gene or DNA. Okay, now when you put it, the first thing happening, it is having the microorganism, the double-stranded DNA of the microorganism. So first thing inside that the temperature will become 95 degree. At 95 degree, the double-stranded will denatured. Both the strands will separate at 95 degree. Can you see? Can you see a break between them? So double-stranded DNA is like this. There are bonds between them. If we are doing the temperature at 95 degree, the bonds will break. And the one double strand will convert into 2 single strand. This is known as denaturation. Denaturation matlab double strand convert into 2 single strand. The bonds get break. All the bonds get break at 95 degree. So first the temperature will become 95 degree inside this. Automatically it will happen. 95, the bonds are broken and the double strand convert into 2 single strand. Got my point? The second thing, as soon as this will happen, automatically temperature will change to 55 degree. You don't have to change it manually. After 95, the temperature will get 55. At 55, what is happening? It contains primer also. So the primer will attach. Now, the 2 strands which are formed from the double strand, this is double strand. Now, it convert into 2 single strand. One is leading, one is lagging. One is leading. So here also primer will come, here also primer will come. The primer, the binding of the primer with the leading and lagging strand is known as annealing. So you can see here also the primer is coming and here also primer is coming. So it is annealing. Denaturation takes place at 95. Annealing takes place at 55. After this, once the primer are done, automatically the temperature inside this will convert to 72. You don't have to change it. So first 95, then 55, then 72. At 72, extension take place. You know adenine form bond with thymidine and guanine form bond with cytosine. We know that. So corresponding, whatever is there, just suppose this is a double-stranded DNA. Okay. First you are doing the temperature 95. So they convert into 2 single strand. Now you are doing the temperature 55. At 55, annealing takes place. Here also primer is binding. Here also primer is binding. And now you are doing the temperature as 72. So at 72, the bonds are formed. Corresponding bonds. So here if it is adenine, here it should be thymidine. Like that? No. Here the corresponding chains, here thymidine, here like this. So the 2-strand convert into the 4 strands. You got my point? So this is extension. This is extension. The last step is the extension. How many of you got it? How many of you got it? So we started with 2 strands and we ended with the 4 strands. So the substrate is the 2-strand, double-stranded. At the end, we got the 4 strands. Now the process is repeated. It's a chain reaction. The product of one is the substrate for other cycle. Now again, the temperature inside this will be 95 automatically. So here all 4 will be separated. Hana, and from 4 to 8. So from 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, so on till millions of copies are not found. And it will take hardly fraction of second to take place. Got my point? Got my point? Huh? You are getting my point. So you are starting with double-stranded DNA. First the temperature is 95. Then the temperature is 72, then the temperature is 55. It's happening automatically. There is a system inside that, automated system. Okay, so here the denaturation take place, here annealing take place, and here extension take place. How many of you got it? So we started with 2 strand, it will end with 4 strand. Now again it will be repeated, it will be repeated, it will be. So from 4 to 8, 8 to 16, 16 to 32, how many of you got it? I would like to show you a video, a small GIF. Hannah, have a look, have a look. I will show you all 3 steps here. Have a look everyone on the screen. See, this is denaturation. The 2 strands are separated. This happens at 95. This is annealing. Can you see the primers are coming? This is happening at 55. And this is extension. Can you see this is happening at 72? This is happening at 72. Hannah, so the 3 steps, So from the 2 strands, you see, these are the 2 strands which got separated. This is denaturation. Again, see, this is annealing, the second step. And see now extension. So we started with 2, we ended with 4. Now these 4 again will enter the cycle. So from 2, we get 4. From 2, we get 4. From 4, we get 8. From 8, we get 16. The process continues. This is known as PCR. You have to put an endpoint. How many copies you want? You started with 2 copies always. And how many copies you want in the end? You want 1,000, 10,000, 1 lakh, whatever. Put an end number. After that, the process will stop automatically. It is happening inside a thermal cycler, which is connected with a computer, with a system, and the software is there inside that. So you have to just put the sample and start. And put the end number. That's it. Okay, and it will happen automatically inside that. Got my point? So this is PCR. How many of you got it? Say, give me a thumbs up. Give me a thumbs up. Do you have any confusion over here? Can we go ahead? Can we go ahead? We will solve some MCQs. The first question is in front of you. Bacterial genes are transferred by means of all of the following except. So which of the following is not a method of gene transfer? I told you 4 methods of gene transfer. Transformation was there or not there? Transduction was there or not there? Mutation and conjugation. I guess the question is super simple. Can you answer in the chat box? What is the correct answer? Of course, mutation is not a way of gene transfer. The 4th way is the lysogenic conversion. You know what are the 4 ways. What are the 4 ways. Mutation is not a way. Definitely mutation is not a way. The second question, bacteria which is used in Griffith Experiment. We do Griffith Experiment to prove transformation. In which we are taking 4 mice, 4 चूहों वाला experiment. But which exact bacteria we are injecting inside the 4 mice? Yes, is it Streptococcus pyogenes? Is it Streptococcus pneumoniae, Streptococcus aureus, or MRSA? That is methicillin resistant Staph aureus. So of course the correct answer is pneumococcus. That is Streptococcus pneumoniae, which is of 2 types, S and R. S is smooth, R is rough. This one is capsulated, this one is non-capsulated. This one is virulent, this one is avirulent. I taught you very nicely. Okay, got my point? Got my point? Okay, how to do the diagnosis, Pradeep? So once the bacteria get the gene of interest get amplified now, different organism have different unique gene. Like the fingerprints. Humans' fingerprint do not match with each other except identical twins. All the bacteria, all the viruses, all the parasites, and all the fungus. These are the 4 microorganisms. Pradeep, these are the 4 microorganisms. All the microorganisms have some or other unique gene which is not present in other. If you are amplifying that gene, and that gene is found in your specimen, just suppose this is my specimen of my patient, and I'm suspecting the patient is having HIV. I'm not sure. I'm suspecting. So whatever gene which is unique for HIV virus, which is not present in other, I will amplify that in PCR. And once it is positive, so diagnosis will be made. So it is used for diagnosis in this way. It is just amplifying. You are right, Pradeep. It is amplifying, but if it will get amplified only if it is present. If it is nothing, if this gene is 0, it will not amplify. So diagnosis is negative. So look for some alternate diagnosis. If it is amplifying, means it is present. It is present. You got my point? This is a way we do it. Okay, so we were doing the questions. If you have any doubt, please write down. Okay, the next question is in front of you. The non-toxigenic strains of Corynebacterium diphtheria is converted to toxigenic strain of Corynebacterium diphtheria by which method? Is it transfection, transduction, conjugation, or lysogenic conversion? I told you Corynebacterium diphtheria is of two types. How to convert non-toxigenic strain to toxigenic strain? How to convert? So for non-toxigenic strain don't have tox gene, and toxigenic strain have the tox gene. So how this will get the tox gene? So beta-phage is going to provide it. Beta-phage, it's a bacteriophage which is going to provide its own DNA. The beta-phage contain tox gene that is given to the bacteria. Okay, so it is lysogenic conversion. It is lysogenic conversion. Please learn it. Okay, movement of DNA from one bacteria to another with the help of a tube or pilus. So the two bacteria are coming physically closer and making a tube between them. I guess easy question. What is the method known as? Is it transformation, transduction, conjugation, or lysogenic conversion? What is the method known as? The two bacteria are coming closer and they make a tube between them. So it is the sex in the bacteria. It is known as conjugation. I told you two types here, F-factor and R-factor. Okay. Now if we change the same question, currently answer is conjugation. Because I am using the word via connecting tube or pilus. If I change the word and if I use via bacteriophage, what is your answer? I am changing the question. Instead of tube, I am using the word bacteriophage. So transfer of DNA from one bacteria to another via bacteriophage. So it is known as transduction. And if I use the word, it is the free or naked DNA which is moving, no agency required, then the answer is transformation. So you get simple questions like that. Okay, so that's all about it. If you want the notes of this session or all the sessions which I have taken on YouTube yet, you can see a link in the chat box which is pinned. So where the chat is starting, you can see a link and that link is pinned. So click on that link which is pinned in the chat box and you will be diverted towards the notes that are free, and all YouTube session notes you will get there in the form of the PDF and you can have a look on that. Okay, it's absolutely free. Okay, and see, here is the QR code. I'm giving you time. If you want to scan it, you can scan it, or you can take a snapshot. You can later on do so. If you are scanning it, you will be diverted towards website, my website, that's MedLive by Dr. Priyanka, where you can connect with me on various social media platforms, that is Instagram, WhatsApp, Telegram. If you wish, you can connect with me, where I daily share multiple, education related material, which is important for the medical students for cracking their exams. Small reels are there, multiple material is there. Okay. Many image-based questions are there. If you want to connect, you can do so. Okay. And here is the contact number. It's 9321001485. You can save this number. It is only for chat support. It is not for calling. So you can WhatsApp there. You can SMS there. You can WhatsApp there. And whatever is your query, you can ask there. If you have any topic in your mind and you want me to take it on YouTube, you can also text that. Okay. So if you have any query, any doubt, anything related, you can do so. Thank you so much. Let me tell you when is my next session. Every Monday, Wednesday, and Friday, I come live on YouTube in the morning, 9:30 a.m. Okay, so today session is done. Monday. The next session is on Wednesday, and after that Friday. So on Wednesday, that is 27th of December, we have a session on sterilization on the YouTube only. And 29th, we are having a session in pathology, healing and repair. Many students requested me to take this topic on YouTube. Many students are finding it difficult. So thank you so much. Thank you so much. Wishing you all the best. And I just wish all of you to have success in your professional and personal life. That's it. Thank you so much. Bye-bye. Wishing you all the best. Okay. So I am just ending the session. If you have any query in your mind, don't hesitate to ask. Thank you. Bye-bye. Okay. If you like the session, don't forget to click on the like button. If you want the notification of all YouTube sessions one by one every Monday, Wednesday, Friday, you can press the bell icon. And please don't forget to share the link of the YouTube sessions with your friends, colleagues, anyone on the globe who is interested to study all these topics in detail with the super simplicity. Thank you. Bye-bye. Wishing you all the best.